Okay, we're looking at question 46 of the biology air practice test. I'm not a big fan of this question, but what it's asking about is trying to find the gene in, like the DNA with the gene for this NAK pump. This is a pump that lives in the cell membrane of certain cells, like your nerve and muscle cells. And we're trying to find DNA for this gene in different cell types. So we're trying to see if this DNA shows up in the nerve cells, in the muscle cells, or other types of cells. And then we're also looking for the protein synthesis aspect. So I wrote this response. Again, I don't like the question. I don't think it's a great, like, well-worded question. I think it's a little bit vague as to what they're trying to get you to say. And I know there's a key somewhere for this question, but I don't care enough to find it, honestly. I'm going to tell you my thoughts about this question, as far as like how I would try to actually answer it. So I don't care what their key says. So part one, it says describe the results scientists should see if they test both cells, and they're talking about a nerve cell and a muscle cell, for the presence of the gene that produces this protein. So the gene means the DNA strand. So part one there, I called that number one. Scientists should expect to see the gene in both types of cells. And not only that, but that gene should be in every cell in that pig's body. Alright, so they took this gene from a pig. They should find it in every cell because the entire genome, all of our genes, are stored in every cell of the body. So that should be in every cell. Part two, it says describe how protein synthesis, that means the process of building the protein, is required to express the gene that makes this protein. So I guess what they're aiming for is what I put down for number two. So even though every cell has the gene in its DNA, the processes that lead to protein synthesis, that's transcription first, that's what makes RNA, and then translation to make the actual protein, are needed for the gene to be expressed. So the gene itself does not form a functional part of the pump, so instead, the code has to be transcribed into RNA and then translated into a protein, which then forms part of the pump. So this is how genes are expressed in every cell. So the muscle cell and the nerve cell are going to actually go through protein synthesis and make this gene function. Other cells, like maybe a cell in your liver, aren't going to do that. Or a skin cell, they're not going to synthesize that protein. They're still going to have the gene, but they're not going to make the protein. So each cell must use these steps in order to allow its genes to be expressed. The genes that are not transcribed and translated are not expressed in those cells. So again, these specific cells, nerves and muscles, they go through protein synthesis for this gene, and that's why this gene functions for those cells. Other cells that don't need that gene, like skin cells, they don't go through protein synthesis. The gene's still there, they just don't synthesize the protein. Alright, that's enough of that one. We're going to move on. This question we have seen in a separate part of the test. It's just in a different sense. You're supposed to decide which things each of these organisms has. So again, if the evolutionary history from beginning to end for each animal, for each organism, has that mark, if it passes through that mark, it's got that trait. So the shark has vertebrae. A bullfrog has vertebrae and two pairs of limbs because it passes through those 2 points. So vertebrae for the bullfrog and two pairs of limbs for the bullfrog. Kangaroo's got three things. They pass through this, this, and this. So vertebrae for the kangaroo, two pairs of limbs, and mammary glands. Last, the chimpanzee's got all four. Vertebrae, two pairs of limbs, mammary glands, and finally a placenta. And those are just like we do. Again, we did this on an earlier question, but it's the same setup. It's just a different style answer. Same answer there. Next up, we also did this one. I'm not sure why some of these are getting fed into this test twice. This is the same question from earlier. And earlier, again, if you didn't watch that video, this is alcoholic fermentation. This is the kind that happens in most of our food options. The other type of fermentation is lactic acid. That's what happens in your muscles. Lactic acid fermentation creates lactic acid. Lactic acid builds up in your muscles. That's why your muscles burn when you work really hard. That's why your muscles are sore the next day. but this one is alcoholic. Alcoholic fermentation makes two main products: carbon dioxide gas. That's why like bread rises. That's why it's like kind of light. That's why cakes rise. It's also why like Swiss cheese gets holes in it, because those holes are where carbon dioxide was collecting inside the cheese. So CO2 is one, and the other one is ethanol. Ethanol is a type of alcohol. Again, it's alcoholic fermentation. This is the kind of alcohol that people drink. So this is how when they brew beer, they ferment their grains. They make wine. They ferment the grapes. They're making when they ferment them, they're producing this ethanol, and that's the part that they use for these alcoholic beverages. The optimum temperature here is based on, again, because CO2 is a product, we're basing that on the highest amount of CO2 produced. So that's this one, that's 35 degrees Celsius, and that one's done. Next up, match the statements with the correct process. So glucose and oxygen are products of photosynthesis. They make glucose during photosynthesis. That's the main thing. Oxygen is a waste. CO2 and water are products. So that's just the opposite. CO2 and water, that's what we breathe out because we go through respiration. Glycolysis happens before respiration. Glucose and oxygen are reactants. These are the things that go into the reaction for cellular respiration. The Krebs cycle, that happens in cellular respiration, and the electron transport chain. For photosynthesis, the Calvin cycle. I think that probably is coming, maybe not. Yeah, I don't see it. Sunlight must be absorbed, photosynthesis. Takes place in the mitochondria, cellular respiration. Chloroplasts, photosynthesis. Produces ATP, respiration. CO2 and water are reactants. Those are the products that go in, or the reactants or the substances that go in for photosynthesis to happen. Those go in. Glucose and oxygen come out. And last, light-dependent stage and light-independent stage, that's photosynthesis. So those are our two processes and what happens for each one. Alright, this one, we've done this in class before. This is set up for, we've got two genes here. One gene is the big B, little b gene that makes the dog black. That's black coat. And little b is brown coat. That's chocolate colored lab. And then there's a second gene, which is big E and little e. If the E is recessive, the dog's coat will be yellow regardless of the alleles for the other coat. So we're looking for we're going to decide what color dogs these are going to be. Good. So that's called an epistatic gene. It will, even though we might see a black or a brown colored coat as far as the B allele, if the E allele is recessive, we don't see any color. So it's going to end up being yellow. It's going to be pale. So this one's going to be black. Most of these are going to be black because as long as we see a big B and a big E, it's going to be black. This one will not. This one will be yellow because there's little e's. This is going to be little e, and that makes for a yellow coat. Black. And there are no brown dogs in this. This one's another yellow one. Black. Black. Hey. This one's going to be yellow again. And then we're back to black here. And I think yellow for the last one. So there is our big old Punnett square. Next up, we're looking at salamanders that live in this California area. What happened is, over time, they started out as a population up here, and some of these individuals headed off to find their own little home. So we established these guys and these guys. Some of them split up here, and then some of these guys moved on and established these, and then some of these guys moved on and established these. So there are actually 5 different populations that came from this original one. And again, you see this divide in the middle. These guys on the ends do not meet up with each other. N and P here, they don't meet up with each other. So they only meet up with their neighbors going along the edge of this ridge. This is a big valley, so these guys don't cross it. So looking at these guys, this question asks you, where do we see gene flow? Remember, gene flow is the movement of individuals from one population to the next. So if you look at these, any populations that are side by side can have gene flow. So this is pretty straightforward. Pick any populations that are side by side. I'll start at the bottom and say P and O. P and O are next to each other. I'm going to pick those two, P and O. I don't know what happened to them. Come on, get in there. There, P and O. Those guys have gene flow because they are next to each other. So those individuals can like cross back and forth between populations. All right, next up, which ones are going to be least likely to reproduce? Again, because these populations started here and they kind of took their own pathways, the ones who are at the most extreme ends or have been the most isolated from each other. So that means P and N are the most isolated from each other. They're on the extremes. P and N is your best answer. Next up, a cladogram. Looking at our map, we see how they split. L and N went to one direction, so L and N are actually, we see a little separate pairing over here. L and N go there. As far as our other group goes, I think they can be, I don't think there's a specific order for them. I would expect M was established first, and then N, so I would put M here, and N and O, or I'm sorry, not N and O, P and O split off from those two at a later date, just because they're further down, so those probably split later. So this is probably, I think, the best answer. N and L could switch, these guys could switch, but overall this is I think the best way for that to go. We go here, we're going to skip this one. We're going to skip this one. They're the same questions. So, and we're going to stop here actually. We're going to do this one on the next video.